Distinși membri ai Academiei de Științe a Moldovei, dragi elevi și studenți, onorată asistență, bună ziua! În decursul acestei săptămâni își va desfășura lucrările Școala de vară Calea spre descoperiri științifice, ajunsă la cea de-a treia ediție, care în anul curent este organizată de Academia de Științe a Moldovei și Academia Tinerilor din Republica Moldova, în parteneriat cu Academia Europeană de Științe și Arte, Academia Română, Academia Științe Juridice din România, cu sprijinul financiar al UNESCO. Pe parcursul anilor am acumulat o experiență bogată în organizarea școlilor de vară pentru tineri. Voi face referință la școala moldo-japoneză în domeniul nanotehnologiilor și ingineriei biomedicale, organizată între 20 și 24 septembrie, 10 ani în urmă, în 2015, de către Academia de Științe și Universitatea Tehnică, pentru tineri din Republica Moldova și Japonia, pasionați de știință și tehnologie. Dacă e să ne referim la seria școlilor de vară cu genericul Calea spre descoperiri științifice, ea și-a luat startul între 13 și 15 iunie 2023, în cadrul proiectului Noaptea Cercetătorilor Europeni. A doua ediție a școlii a avut loc anul trecut, între 27 și 31 august 2024, fiind organizată în comun de către Academia de Științe a Moldovei, Academia Română și Academia Tinerilor din Moldova. Tradițional, în cadrul școlilor de vară conferențiază experți notori din diverse țări ale lumii, inclusiv laureați ai Premiului Nobel în diverse domenii, președinți și vicepreședinți de Academii Europene și naționale, etc. Ediția a treia a școlii de vară, la fel ca și edițiile precedente, poartă un caracter pur multidisciplinar. Ea se va deschide astăzi de profesorul James Rothman de la Universitatea Yale din SUA, laureat al Premiului Nobel în medicină, prin susținerea prelegerii de ce este importantă știința fundamentală. Va urma prelegerea inteligența artificială susținută de domnul profesor Klaus MAINZER, președinte al Academiei Europene de Științe și Arte din Salzburg, după care academicianul Aurelian GULEA va conferenția pe tema perspectivele de tratament al cancerului. Diverse aspecte ale științelor medicale vor fi dezvăluite în decursul școlii de vară de către academiciană Eva Godomac, vicepreședinte al Academiei de Științe a Moldovei, profesor Irina Iuliana Costache-Enache din Iași, doctor Iuliana Popescu din Statele Unite, membru corespondent Liliana Gropa, profesorul Nicolae Calea și profesor Viorel Nacu de la Universitatea de Medicină și Farmacie Nicolae Testimițanu. Domeniul biotehnologiilor va fi reflectat în prelegerea membrului corespondent Liliana Cepoi de la UTM. Domeniile fizicii, chimiei și ingineriei vor fi prezente în prelegerile susținute de către academicianul Juras Banis, președinte al Academiei de Științe din Lituania, academicianul Marius Andruh, vicepreședinte al Academiei Române, membru corespondent Viorel Bostan, rector al Universității Tehnice, profesor Xiao Yuang din China, profesor Andrei Rotaru din România, profesor Cornelius Nils din Germania și alții. În domeniul științelor juridice vor conferenția profesor Liviu Bogdan Ciucă, președinte al Academiei de Științe Juridice din România, dr. Marian Enache, președinte al Curții Constituționale a României, dr. Stephanie Boyce din Marea Britanie și Robert Williams din România. Academicianul Alexandru Stratan, rectorul ASEM, va descrie cum economia modelează cariera și viitorul tinerilor, iar profesorul Violeta Dinescu, de la Academia Europeană de Științe și Arte, va dezvălui cum se scrie o operă pentru copii. Un alt reprezentant al Academiei Europene, profesor Ioannis Liritzis, va expune cum interacționează domeniile STEM, asta înseamnă știință, tehnologie, inginerie, matematică, cu artele. Domeniul agriculturii va fi reprezentat de profesorul Ratan Lal din SUA, laureat al Premiului Nobel pentru Pace și al Premiului Mondial pentru Alimentație, precum și de membru corespondent Boris Boincean. Școala de vară, ediția anului 2025, se impune ca un eveniment multidisciplinar inedit, cu o participare largă a experților din Republica Moldova și de peste hotarele ei, România, Germania, SUA, Lituania, Austria, China. La școala de vară participă 36 de elevi cu prezență fizică în sala azurie, precum și mulți elevi online, inclusiv din România, pe platforma Zoom. Distins auditoriu, vreau să vă atrag atenția la faptul că astăzi este o zi deosebită și din alt punct de vedere. La 23 iunie 1990, acum 35 de ani, a fost aprobată Declarația Suveranității Republicii Moldova, care peste un an, la 31 august 91, a proclamat independența. Îmi permit să dau citire la câteva aspecte ale acestei declarații de suveranitate, recunoscând adevărul că toți oamenii sunt egali și au dreptul inalienabil la viață, libertate și prosperitate, conștienți de responsabilitatea istorică pentru destinul Moldovei, ce are istorie, cultură și tradiții milenare, respectând dreptul la suveranitate al tuturor popoarelor în scopul instituirii justiției, ocrotirii legalității și stabilității sociale, exprimând voința poporului, proclamă solemn: Republica Moldova este un stat suveran. În textul declarației de suveranitate au fost enumerate drepturile cetățenilor și ale noului stat, care au stat la baza elaborării noii Constituții. Părinții și bunii voștri, ai tinerilor care aveți acum posibilitatea să frecventați cursurile școlii de vară, să plecați liber în statele europene, să aveți acces liber la educație, sănătate, s-au încadrat atunci în mișcarea de eliberare națională și au stat la baza acestei declarații de suveranitate de la care a pornit statul independent Republica Moldova. Mulți dintre ei erau de vârsta voastră. Aveți și voi oportunitatea, împreună cu toți cetățenii Republicii Moldova, să înscrieți o pagină fundamentală în istoria țării prin implicarea activă în procesul de aderare a Republicii Moldova la Uniunea Europeană. Pentru a celebra curajul înaintașilor noștri și aspirația noastră comună de a trăi liber și demn în Republica Moldova, cu prilejul împlinirea 35 de ani de la adoptarea declarației de suveranitate, astăzi, la monumentul lui Ion și Doina Alde Teodorogici pe strada Alexei Mateevici din Chișinău va avea loc marșul, ce reprezintă un moment de celebrare a primei mari victorii în lupta pentru renaștere și eliberare națională. Voi reaminti că exact 3 ani în urmă, pe 23 iunie 2022, Consiliul European a recunoscut perspectiva europeană a Republicii Moldova, acordându-i statutul de țară candidată pentru aderarea la Uniunea Europeană. Onorată asistență, în numele prezidiului Academiei de Științe a Moldovei, vă exprim sincere felicitări cu prilejul celor 35 de ani de suveranitate a Republicii Moldova. Rog să fie interpretat imnul Republicii Moldova. Vă mulțumesc. Onorată asistență, cuvânt se oferă domnului membru corespondent Emil Ceban, președinte al Consiliului Rectorilor din Republica Moldova. Domnul membru corespondent, vă rog.
Bună ziua tuturor! Onorate domnule președinte al Academiei de Științe Republicii Moldova, domnul academician Tighineanu, onorat prezidium, stimați academicieni, stimați membri corespondenți, stimați colegi, dragi tineri, în primul rând sunt aici ca să vă mulțumesc pentru că ați ales școala de vară organizată de Academia de Științe a Moldovei și Academia Tinerilor din Republica Moldova. Și vreau să vă mulțumesc că sunteți astăzi într-așa o zi semnificativă pentru țara noastră, pentru viitorul Republicii Moldova, pentru independența Republicii Moldova și a coincis cu organizarea a 3 ediții a Școlii de vară. Astăzi, tineretul studios se dezvoltă. Universitățile, eu prezint președintea Consiliului Rectorilor de Universități din țară, dumneavoastră sunteți viitorul nostru, sunteți cei care de pe băncile liceilor, deci într-un an, 2, următorii ani, ați fiți, deci, membri a comunităților academice a universităților din țară. Astăzi universitățile se dezvoltă vertiginos și în strategia de dezvoltare a universităților este și cercetarea și inovarea ca un punct foarte important. Ani de zile acest lucru nu a fost pus în atenție și ultimii ani Academia de Științe a Republicii Moldova, Consiliul Rectorilor în Republica Moldova, împreună cu Ministerul Educației și Cercetării, deci au pus în strategie de dezvoltare a universităților cercetare. Școlii de vară este un instrument modern de a participa și a învăța lucruri noi. Mă bucur faptul că tematica acestor școli de vară, mai ales tematica de anul acesta, este multidisciplinară și este foarte, foarte bine aranjată. Deci și experții care să aveți onoarea să-i ascultați sunt lideri mondiali, sunt lideri naționali, sunt personalitățile notorii, deci din cercetare. Și acest lucru, deci dumneavoastră o să puteți vă familiariza cu cele mai noi lucruri ce se face astăzi în știință, în cercetare. De aceea sunt aici ca să vă mulțumesc, sunt aici să vă rog să faceți cercetare, să priviți la cercetare cu alți ochi, cercetarea să fie indispensabilă pe lângă educația și învățarea pe care o faceți dumneavoastră. Știți că un viitor nu poate fi fără cercetare. Și iată, la Academia de Științe a Republicii Moldova, aici este locul unde cercetarea stă în capul mesei, unde cercetarea este un lucru indispensabil. Și voi aveți norocul mare să fiți în sala azurie, unde academicienii, personalitățile notorii care contribuie la dezvoltarea cercetării în Republica Moldova, iau cele mai importante decizii. De aceea, această sală, sala azurie, este foarte importantă și aș vrea ca să o memorizați și poate, de ce nu, voi, peste ani de zile, să fie viitorul nostru, să fie cei care să fie în acești fotolii și îți aduce cele mai importante descoperiri pentru dezvoltarea țării noastre. Eu vă mulțumesc pentru acest lucru și vă doresc succes și o școală de vară, prelegeri cât mai interesante pentru dumneavoastră, cât mai multe descoperiri, cât mai multă atragere către cercetare și inovare. Într-un ceas bun.
Vă mulțumim mult, stimate domnule rector. Dragi elevi, voi modera astăzi Școala de vară împreună cu doamna academician Eva Godomac, vicepreședinte al Academiei de Științe a Moldovei, și avem în prezidiu și domnul academician Aurelian GULEA, care va prezenta a treia prelegere astăzi. Prima prelegere va fi prezentată de domnul profesor James Rothman din Statele Unite ale Americii, Câteva cuvinte despre domnul profesor. S-a născut pe 3 mai 1950 în New York, Statele Unite ale Americii, a studiat biologie la Universitatea Columbia, unde a obținut diploma de licență. Ulterior, și-a continuat studiile la Universitatea Harvard, unde a susținut teza de doctor în biochimie. De-a lungul carierei a fost profesor la Universitatea Yale, conducând laboratorul de biologie celulară. Profesorul James Rothman este un biolog și biochimist remarcabil, cunoscut pentru contribuțiile sale în domeniul biologiei celulare și al transportului celular. În anul 2013 a câștigat Premiul Nobel în medicină, împreună cu profesorii Randy Sheckman și Thomas Sudhoff, pentru descoperiri legate de mecanismele de transport celular. Profesorul Rothman a studiat modul în care veziculele transportă proteinele și alte molecule în interiorul celulelor, contribuind semnificativ la înțelegerea funcționării celulelor vii. Astăzi veți savura prelegerea domnului profesor, am pregătit-o și va fi și subtitrare, dar pe secret vă spun că domnul profesor Rothman în octombrie ne va vizita pe viu și o să vă invităm și la prelegerea care va fi prezentată în octombrie. Domnul doctor Tudor Braniște, vă rugăm să puneți.
Well, good morning or afternoon to all of you. I'm not sure what time of day this video is being shown, but in either case, I'm very happy to be with you for your summer school. I'm sorry I can't do it in person, but I've heard a good deal about what you are doing, and certainly I'm very supportive of your consideration of a career starting at the university level in science, in scientific research, technology, or in related areas of what increasingly we call STEM. And what I'd like to do today is offer you sort of my perspective, which has been gained over approximately, well, almost half a century, I'm embarrassed to say, of research at the university level in basic science, in biology, and in medicine. And I believe after all of these years, I'm only more convinced that basic research is important. And what I'd like to do is share with you with a few examples from biomedical research in particular, why I feel that way. And I hope that you'll consider the importance of basic research for our societies, our lives, and our livelihoods when you consider the career path that you may take. Now, although I will emphasize today examples from biomedical research, I could equally well be talking about basic research in material sciences, engineering, computer science, chemistry, and the list goes on. My personal competence, though, limits me to talking about biomedical research. So let's begin with a fact that many of us are not aware of. This is a graph of life expectancy, human life expectancy, going back to some of the earliest recorded history. So if you go back to the, let's say, the Roman era, the classical era, the life expectancy at birth was about 25 years. Actually, as far as we can tell, although the records are poor, you can go back to the earliest recorded histories, and it appears that the life expectancy was no different. But if you look at this graph, you'll see, astonishingly, that nothing changed up until about here, that being the middle to the late 19th century. So what happened in the middle or the late 19th century that caused a dramatic increase of life expectancy which now globally is in the range of 70 or more years. What happened? Well, what happened was the beginning of systematic basic scientific research in medicine in the mid-19th century. We all think of the name Pasteur, for example, or Koch, famous French and German and other European names of medical scientists who took the scientific approach, the scientific method, which was first developed, as you probably know, for the study of physics and the physical world, and then the chemical world, and took this method of thinking and of evaluating the world into the world of biology. And what they discovered very quickly was that common diseases were caused by microorganisms that were invisible to the eye, and they developed ways to measure these microorganisms and to quantify them. Methods of microbiology that we still use today, as a matter of fact. They discovered that these microbes were present in water in most cities, and that contamination of the water, and as is still the case in many parts of the world today, unfortunately, is a prevalent cause of infectious disease. They learned and taught us how to treat water with, for example, with chlorine, and how to carefully dispose of water and waste, which all big cities endeavor to do today. Health departments were instituted in various countries throughout the world, and the immunologist, soon with specific microorganisms already now associated with causes of diseases, the field of immunology could develop, and with this came the systematic approach to developing vaccines, which of course we all benefited from even recently during the COVID pandemic. And you can see here, this is now the death rate due to infectious disease as a function of years in the 20th century. You can see some of the major events, a decline in the incidence of infectious disease or in the death rate from infectious disease by chlorination of water, a simple public health maneuver. By the way, in the 19th century, the greatest advance in public health was made by Semmelweis, a Austrian-Hungarian physician who discovered that microorganisms could be their contamination minimized by a simple act of hand washing. And that's, of course, saved many lives. But the development of vaccines, for example, after the influenza pandemic, influenza vaccines, the discovery of antibiotics, very importantly during my lifetime, the Salk vaccine, as we call it, for polio, was introduced, and you can see there was a dramatic decline. So this was one of the first victories of basic medical science, the discovery that diseases are caused by specific microbes. Now, what was the consequence of this? Well, if people were dying in childhood or early adulthood as a result of infectious disease, and unfortunately we haven't completely stopped this yet, but we've certainly put a huge dent in it. If people die less from infectious disease, well, they live on. And when people live longer, what happens? Well, I think unfortunately we know what happens. We start to see degradation of the cardiovascular system, the blood vessels, the heart, the circulatory system, and with that, the development of atherosclerotic disease, strokes, heart disease, and the like. And so, perhaps not surprisingly, as deaths due to infection went down during the first half of the 20th century, now people live longer into their 50s or 60s. And good news, life expectancy went up, as I've been saying. But bad news, people lived long enough to develop, on the average, cardiovascular disease. So what happened then is that the incidence of cardiovascular diseases, whether they're diseases of the heart, this is now again death rate, not the rate of incidence of the disease, death rate per 100,000. These are US numbers, but they are reflective of really any developed society in the world at that time. Cardiovascular disease went up, as I've described, due to the heart and also due to stroke. But then you see something else happen. Look what happened starting around 1960 through about the mid 1990s. Look at total cardiovascular disease deaths. There's an enormous decrease. It's more than a factor of 2, and by today it's actually a factor of 3. And again, this is something that has been kind of the story of my generation. My parents and people of my generation, our parents, were approaching their mid-50s in the 50-year-old or greater in the 1960s, 70s, and 80s. I'm a member of the so-called baby boom generation, born after World War II. And so as we started going, when I was essentially your age, our parents were perhaps like your parents in their 40s, 50s, and 60s. And they were experiencing cardiovascular disease. I well remember, happily not my parents, but I well remember many of my friends' parents, especially the men, simply dying of heart attacks in their 50s and in their 60s. In fact, it was uncommon for a man to live beyond about 70. The life expectancy then was more like 65 to 70. The retirement age then as now was mostly about 65. But when a man retired and it was mostly men in those times who were employed, unfortunately, but it was the case. And when men retired at 65, they actually had a rather short retirement on the average to worry about. Happily today, when people retire at 65 or 70 or older, they typically will live much longer than that and have an opportunity to develop new interests and new careers or to spend extended time with families. And why is that? Why is it that men, and of course women too, women have always had perhaps 5 years more life expectancy than men on the average? Never been completely clear why, but it's also true here for cardiovascular disease. So why is it that this happened? It's not an accident, actually. It's not like suddenly everything changed, the world changed in 1960, and suddenly people started getting spontaneously cured or treated for heart disease. So what happened? Well, this is what happened. We came to understand that the underlying causes of atherosclerosis were high blood cholesterol and high blood pressure. Those two factors together conspire over many years to lead to the deposition of atherosclerotic plaques at critical places in our vasculature that affect the integrity of the vasculature in such a way as to weaken it, to allow bleeding, which is what we call a stroke, particularly when it occurs in the brain, or to cause blockage through fatty cholesterol plaques that are released from where they accumulate in one part of the vasculature typically, or even can grow in situ to block our coronary arteries in the heart, or arteries the arteries that go to the brain, or other critical parts of the vasculature. And so what happened starting in the 1960s is, as the result of deep understanding of physiology, basic physiology, we came to understand blood pressure and the basis of controlling blood pressure. And we came to understanding cholesterol as a biochemical entity. We took scientists in that era undertook a study of cholesterol for its own sake. They were interested in what composed an atherosclerotic plaque. They discovered cholesterol in the plaque. And there began 50 years of study, as I'll describe now, of the basic biology of cholesterol. And what that translated into are drugs that we take today that control our cholesterol. I personally have had high blood pressure and high cholesterol. And I'm now 74 years old. I doubt that I would have lived a lot longer than my grandfather, who came from Moldova, actually, who died, unfortunately. I never got to meet him, in his mid-50s. And he died in New York, but he died of a combination of high blood pressure and cholesterol, as many people did in that era. He never had the benefit of taking a drug for blood pressure or a drug for cholesterol, which I do. Now, what is the story then behind the development of these major discoveries, and how did they occur? Now, I won't have time today to go through both of these stories, so I'm going to pick the cholesterol story. But what I say about cholesterol could equally well apply to the discovery of how to control blood pressure. They're both equally important for controlling heart disease and stroke. And importantly, and this is really the major lesson today, importantly, what we learned from cholesterol applies really to any basic science program, and the impact that basic science can have, and why it requires long-term investment and a lot of patience over many years, many more than we would like to wait for these types of advances, but it's the nature of science. So where does this cholesterol story begin? Where did the drugs come from that we use today to control cholesterol and save lives on an epic scale? The story actually begins in the early, around 1920, even a little before 1920, in the wake of the recovery from World War I. And even by then, European science, especially Germanic science in medicine, was operating. When I say Germanic science, I'm referring primarily to science in Switzerland, Germany, Austria, to some extent, Hungary is part of the Hungarian Empire. And I can't remember if I mentioned Switzerland, but I probably did. In any case, these scientists, Heinrich Wieland and Adolf Windhaus, were recognized by a Nobel Prize for their discovery of the structure of cholesterol. They were just curious to know these atherosclerotic lesions, which in their time had already been associated by pathologists with heart disease. You could see patients that died from heart disease had abundant plaques. It wasn't hard to find them. And these plaques were composed of a fatty substance. What these scientists and their contemporaries did, they were chemists. They were natural product chemists, and they used their skills in analytical chemistry to describe the exact chemical structure of cholesterol. Now, and they described it as the major constituent of these plaques. So it became quickly important, and they postulated that cholesterol was important for heart disease. And they provided the first way of proceeding in a scientific basis. They reduced it to a chemical problem. They now had a hypothesis. The hypothesis is, for some reason, cholesterol accumulates in plaques. They discovered the structure of cholesterol, and now they opened the door to a new problem. We could now ask, or they could ask, how is cholesterol made in the body, and how is it absorbed from the food that we eat, so that that combination of the two could determine how much cholesterol you have in your body, and possibly even where it goes in the body, maybe even into a plaque. Well, they also discovered something else. They characterized, for example, bile acids. Now, that doesn't seem like they would have anything to do with cholesterol. They are the secretions of the bile duct of the gallbladder. And some of you may know that the gallbladder stores these compounds called bile acids, and it injects them into the gastrointestinal tract to facilitate the digestion of food, okay? And really what the bile acids are they're sort of soap-like molecules that act as detergents to dissolve the food, so that the enzymes in our digestive tract can eat them up and break them down to substituents, small molecules like amino acids or fat fatty acids very effectively. But it turns out the bile acids are metabolites of cholesterol. They actually are simple derivatives of cholesterol, and the relationship between the two was also discovered by these two scientists. And then when those two classes of molecules were put together side by side, it could be seen that there were certain structural relationships. And that enabled, over the following years, these two scientists working in Germany and in Switzerland to come in from a completely unrelated way to discover yet another set of molecules that had something in common with cholesterol and with bile acids. And these are the sex hormones. Now, I think we've all heard of estrogen and testosterone as kind of the major so-called sex hormones that determine male or female, okay, and are important for many other aspects of biology and medicine, and are also very important in prostate cancers, breast cancers, and the like. Those discoveries actually go back to these scientists who wanted to identify, were simply asking, were there hormones that could be isolated from the blood or the urine that were unique to males or females and then might have a determining role? And so they discovered testosterone and estrogen, and they discovered when they analyzed the structure of these molecules, they saw that actually they had a very similar structure to the structure of cholesterol and of the bile acids. So suddenly the world of cholesterol expanded very broadly into a world of multi-cyclic, heterocyclic chemistry of fatty substances that were related to cholesterol, steroid hormones. And they also noticed a similar structure was present, believe it or not, in rubber. Rubber that was being of great importance, being withdrawn from plants in exotic climates, and being used at that time primarily in industry for the newly expanded industry of automobile tires, and of course for many other things. And what they discovered was that all of these substances, cholesterol, steroid hormones, were made from a basic repeating 5-carbon structure that is called an isoprene. And this, I'll show you an example of that structure in a little bit. I'm trying to avoid showing you a lot of chemical formulas. But I hope you're getting the feeling that an understanding, and so I want to go back a step. The question that we're trying to ask is, how is cholesterol built in the body? The problem at that time in 1930, 1939, is that we actually didn't know how any carbon-containing molecule essentially was built, macromolecule was built in the body. We knew a little bit about how food was metabolized. For example, you've probably heard of the pathway of glycolysis. Okay, but we knew very little about how the body built up substances. So the clues were coming from many different sources. Now I'm going to take a detour, which is completely wild. It's 15 years later, and it's the United States, and this is a German scientist, actually, a student of Wieland, who has one of the men who discovered the structure of cholesterol, who's now emigrated to the United States and is asking an even more fundamental question. So I hope you understand what's happening here and can appreciate the questions are getting more and more fundamental as we try to answer a question for a medical reason. What is cholesterol, the substance in the plaque? Oh, it's this 5-ring compound with a little aliphatic tail. I'll show you the structure in a bit, in case you don't know it. And in order to answer that question, you had to figure out how the cholesterol gets made. But to do that, you have to figure out what it looks like as a chemical. And then you suddenly notice that all these other clues are coming from other areas of biology and medicine, completely unrelated, going from plants to people. And you realize that there are certain commonalities of the chemical structures. And those commonalities are actually clues as to how the molecule of cholesterol is put together. I'm going to give you a hint. It's later we're going to find, many years later, that from discovering the pathway of cholesterol synthesis, how it's put together, that's where the drugs came from, because the drugs prevent the synthesis of cholesterol. So all of this had to happen for those drugs to be developed. But now in the 1940s and 1950s, we see Fritz Lippmann has actually made a very important discovery. He's discovered how it is that carbon bonds are made in the body. This is maybe the most fundamental discovery, one of the most fundamental discoveries in all of biology and biochemistry. How is energy utilized to activate a carbon molecule so that it can be linked to another carbon molecule? Because I think you all know that the core feature of human biochemistry and any biochemistry is the organic molecules that comprise our bodies that have carbon-to-carbon bonds. And it was Fritz Lippmann who discovered a molecule called coenzyme A that is the key for activating virtually every carbon-to-carbon bond biosynthesis in the body. And he discovered coenzyme A's role in relation, actually, to an unrelated biosynthetic process. But this then introduced the concept that quickly migrated back across the Atlantic Ocean to the laboratory in Munich of Theodor Lunin, and also the laboratory in the United States of Conrad Bloch, who was at Harvard in Cambridge, Massachusetts. Theodor Lunin was at the Maximilians Universität in Munich, and a member of the Max Planck Institute. And what Bloch and Lunin independently discovered, now armed with the idea that coenzyme A, coming from Fritz Lippmann, the idea of coenzyme A activating bonds, carbon-carbon bonds, that was the key clue that enabled them to discover the mechanism of cholesterol synthesis. And so I'm now going to actually show you some chemical diagrams. And what we're looking at here is this 5-carbon molecule, which is that I've been mentioning, this 5-carbon intermediate that is the basic structure that will be made into cholesterol. And it's called HMG-CoA, hydroxymethylglutarate coenzyme A. There's coenzyme A. And what coenzyme A does is basically it makes a high energy bond to a 2-carbon acetate unit that then allows this 2-carbon acetate unit to be added with other 2-carbon acetate units into what amounts to a polymer. And that polymer, in some cases, is limited to 5 carbons, and it's further modified, and is then used as the basic ingredient, adding one after another, to make the core structure of cholesterol and the core structure of rubber, terpenes, the core structure of steroid hormones or of bile acids. So it all comes from this one enzyme reaction. And the enzyme that carries out this reduction of mevalonic acid, the precursor, which comes from acetyl-CoA to acetyl-CoA to HMG-CoA to mevalonate, and then is further metabolized into cholesterol and the related compounds. This turns out to be the key regulatory enzyme. And so if you could inhibit this enzyme, you would then inhibit the pathway of cholesterol synthesis. And actually, that was the discovery made by Michael Brown and Joseph Goldstein in Dallas, Texas, at Southwestern Medical School in the early 1980s, and was awarded the Nobel Prize in the mid-1980s. And they discovered the enzyme, the key enzyme that is the regulatory enzyme that I just mentioned, HMG-CoA reductase. And importantly, they discovered that this is the enzyme that is the regulatory enzyme in human beings that controls the amount of the body cholesterol. They also elucidated a pathway that I won't go through in detail that made it very clear that if you could inhibit this enzyme here with a drug, HMG-CoA reductase, that drug would work to lower cholesterol in the body. And so that then enabled a Japanese scientist whose name was Endo to screen fungi, okay, much as Fleming and others had screened fungi to discover penicillin, now and therefore to develop the first antibiotic. Endo in the early 1980s screened thousands of strains of fungi to identify a few that secreted a molecule that very potently inhibited HMG-CoA reductase, and therefore, according to Brown and Goldstein, using the pathway of Lunen and Bloch, which was based in turn on the discoveries of Lippmann and Lunen, and the earlier discoveries that I've mentioned to you of the structure of cholesterol that enabled all of this, he was able to discover the prototype drug Endo did for what we now call statin drugs. They were further developed in the 1980s by the American pharmaceutical company Merck, and the first was approved in 1987. And so maybe you remember that we saw this dramatic drop in cardiovascular disease, and that a lot of it has occurred from 1980 on. This part of the curve was blood pressure, and this part of the curve are the statin drugs. And the story of the statin drugs then, I just went over for you. And I hope you can see that more than half a century of basic science resulted in the development of these drugs. Beginning between 1910 and 1920, the structure of the cholesterol molecule, the understanding that the structure must have a very broad biochemical basis that has in common, that takes into common, that includes the biogenesis of rubber. Rubber is a compound which is a direct repetition of these 5-carbon isoprene units. The discovery that these isoprene units are made from acetic acid and made using coenzyme A for activation, and their biosynthesis was uncovered by Bloch and Lunen in the 1950s, taking advantage of the discovery of coenzyme A and its even more fundamental role, an entirely curiosity-driven discovery by Lippmann made in the late 1940s. And this led to Brown and Goldstein discovering through the genetics of certain families of humans who have constitutive high blood cholesterol. They discovered the gene that was important for that. And they discovered that that gene controls HMG-CoA reductase. And then that opened the door for the pharmaceutical discovery, initially in the university in Japan, by Endo of the first prototype drugs, which were then further developed and made manufacturable and synthesized by Merck and Company, and later by every major pharmaceutical company. So the lesson, I think, to take away from this, and I'd like to emphasize to you today that I could be talking about really any field of basic science. The lesson is that basic science can have profound, practical impact. Look at the example of cholesterol in saving lives even today. Where did that impact come from? It came from basic science that was broad, deep, and enduring. You know, if the government had decided, any government in let's say 1920, we know cholesterol is there in plaques, we're going to start a program, and we're going to make this the decade of cholesterol. And we're going to invest $1 billion in solving cholesterol. You know what would have happened? They would have spent $1 billion and they would have had nothing for it. But they could have said something quite different. We're going to spend $1 billion over the next 10 or 20 years or 30 years, and we're going to enable individual scientists, just as you saw here, to follow their curiosity and to go about in their own way to understand deep problems of enduring value, basic problems. How does the body make a carbon-containing compound? How does the body make any compound? And so scientists who were well-funded in programs that were driven by their own initiative, rather than top-down targeted programs, succeeded. Admittedly, it took some time, but they succeeded not just in solving cholesterol, but in many other important advances that were made. And so today, the lesson that I think we all should learn is that funders should invest in great scientists, especially young great scientists with great promise, not necessarily great projects or great goals. The great goals will come from funding great scientists. There are many other examples of that, and a current one, which I won't go through in any detail, is the enormous progress that we're making in cancer. Okay, I think it's no secret that that there are many, many wonderful treatments for cancer today that didn't exist 20 or 30 years ago. And what they're based on is the single most important discovery in cancer biology, in my opinion, which was made by these scientists in the 1980s. And they discovered that cancer is a genetic disease. Sometimes people are born with genes that predispose us to cancer, but more often these cancer genes are acquired by mutation in the body, a so-called somatic mutation of genes that normally function very positively and favorably in the body, but can malfunction when they're mutated to cause uncontrolled growth and metastasis. That fundamental discovery was further enabled by the description of the human genome. Around the year 2000, we had our first complete human genome. It cost billions of dollars to get the sequence of that human genome. Now, it's a matter of hundreds to $1,000 to routinely sequence the genome of any one of us. And as a result of this democratization of the genome, beginning around 1920, there are many, many, many genes that have been discovered and connected to cancer. And they've been connected to cancer through fundamental research, not just directed at cancer, but research, including, I have to say, my own research in molecular cell biology, which governs cell division, communication, and physiology. And we still continue to see many new drug targets. And over the last several years, this combined with high throughput methods for drug discovery and the development of monoclonal antibodies has given us really dozens and dozens and dozens of very important new treatments for cancer. And there are drugs and treatments that have been approved that really target every aspect practically of what goes wrong in cancer, whether the targeting is through pills that you take or injectables, monoclonal antibodies that are directed against the cause of disease. Most recently and importantly, there are drugs, a class of drugs called checkpoint inhibitors that are monoclonal antibodies that take advantage of our understanding of basic aspects of the immune system and allow the immune system to be unleashed for controlling, unfortunately not all, but many cancers. And even as we speak today, we're seeing the development still in clinical trials and often at the university, not yet in pharmaceutical companies, of specific cancer vaccines that are taking advantage of characterizing the genome in an individual cancer and then developing individualized vaccines for the very specific alterations or mutations in one individual cancer. And so we're starting to see bespoke, individualized cancer vaccines that can work today that couldn't work before because we have checkpoint inhibitors that release the immune system and now specific personal vaccines that redirect the immune system according to the nature of the individual tumor. We're not yet seeing the clinical effects of that on a large scale, but it seems very clear that we will. And when we have, we will see the fruits of nearly half a century, starting about the 1960s, of molecular and cell biology harnessed in the most effective way. And looking forward, even beyond that, we have one of the most profound discoveries that was made very recently, between 1910 and 1915, of genome editing, which is allowing us to do genetic surgery. And we've seen the first drug today that has been clinically approved for specific genetic surgery to change a base within a patient in a targeted fashion to correct a very specific, admittedly very rare disease. But we will see more and more of this, and in the decades ahead, in which if any of you become practicing physicians or scientists doing biomedical research, or just living your lives as everybody else does if you choose not to go into science, you will benefit from everything that I've said, from the basic science that has continued to develop. And so I've to summarize then, I've sort of taken you through a piece of basic science which has come, I would say, to full fruition. Discovery of the way the body handles cholesterol and the way the body controls blood pressure. And we've seen, if not the full benefit, close to the full benefit for public health in heart disease. With that success, now after that, cancer becomes an even bigger problem. Why? Because people live longer. And we have seen already, and we are now developing the full clinical impact of 50 years of investment in basic cell and molecular biology, and the further development of tools in the pharmaceutical industry that have allowed new cancer drugs. And that still continues and will continue for the next 10 or 20 years. And I'm suggesting that we are at the very beginning of another era of genetic medicine that has come out as a result of the many methods that are now under development, initiated by Charpentier and Doudna and others, for genetic surgery. And we will probably see the full impact of that as in the fullness of your life on neurodegenerative disease, which is probably the most important frontier. We're making an impact on cancer. We know how to do it. We don't know how to do it in every case, but we know that we can do it. And so that means that over time, cancer will be controlled to the point that, if not in my lifetime, in your lifetime, it will be controlled and can be thought and probably described as curable. But today we have an imposing problem. And I'll leave you with this to think about, because as infectious disease has been controlled, as cardiovascular disease has been increasingly controlled, as cancer, and I would say we're at the midway mark, we can give ourselves a good report card, but we haven't yet controlled it, but we're making progress. All of these diseases will be controlled in your lifetime. And what will be the result of that? People will live even longer. And we're already seeing the result of that. What is the one cell in the body that actually doesn't divide, or only rarely does, and so we have to make do with it for our whole lives? It's called the neuron, okay? And we can't get rid of and turn over all of our brain cells, because if we did, we wouldn't have any memory and we couldn't learn anything. So we're really stuck, okay? So we have to find, what happens then is our neurons start accumulating pathology over time. And we know something about the pathology. We don't know enough yet. There's a lot of basic science that needs to be done. And we're seeing a lot of billion-dollar programs that are directed, just as I warned you we shouldn't do. This is the decade of the brain. We're going to get Alzheimer's disease this decade. We're not going to do that, I'm afraid, unless we really understand a whole lot more about the cell biology of the neuron and the basic biology of the brain and how the brain works. And of course, that's going on, and it's a pressing problem. It's estimated that today, 50%, fully 50% of children born today, babies born today, will develop profound neurodegenerative disorders sometime in their lifetime. And we're starting to see that already. It's very clear. It's becoming a public health problem on an epic scale. So honestly, if you look left and you look right, half of the people or more, probably more, of your friends and colleagues today, half of you are going to eventually develop neurodegenerative disease. And so we've got to do something about this to prevent it or to treat it. And that's going to be your biggest challenge if you go into biomedical research. And it's going to be the biggest challenge of basic research to understand. And probably the answers are not going to come from specific studies, frankly, on neurodegenerative disease. I also suspect they won't come from studies of the brain. They may well come from studies of the liver, when we learn what causes oxidative damage in the liver, for example, okay, or the kidney, or maybe from a plant cell. I really don't know. Nobody knows. We need to invest in curiosity-driven research. And so I have one lesson in science that I think that took me many years to appreciate. I used to think that the scientists I admired were, like incredibly smart, that they had something special going, that they knew some secret that I didn't know, okay? And then it turned out over time that I became a successful scientist. So for a while there, I thought I must be very smart, and I must really know some things that are very special that maybe other people don't know, because I made a couple discoveries too, okay? Now, what I finally discovered, though, is that is the best secret of all, and it's not a secret of any kind. It's the scientific method, because there was only one really great discovery made in the history of science, and that is the scientific method. It's a way of approaching the world. It's an empirical way of approaching the world, and it was first applied in physics, beginning with Galileo and Descartes and Newton and others, and later applied over and over and over again. And there is no more important discovery or as important discovery that has ever been made. And so if you just stick to the rules of the road and you keep your emotion out of science, which is, of course, difficult to do if you're a practicing scientist, but you can develop the tools to do that, and you simply apply the scientific method, it has endless potential. And it has led to enormous improvements in human longevity, a reduction in suffering, and I believe will lead to the control of neurodegenerative disease in your lifetimes. And I'll leave you with just one thing, a quote. This is a quote from Edvard Buchner. Most people don't know who Edvard Buchner is. I've made a study of him because he's sort of a hero of mine. A German biochemist of the late 19th century, who actually invented the field of biochemistry. He discovered that yeast, simple yeast, can ferment sugar when they're dead. You can actually, he discovered, take yeast cells that are alive, grind them up into a pulp, and now as a cell-free extract, they will metabolize sugar as rapidly or more rapidly as a living yeast cell. And in so doing, he discovered enzymes. He discovered the way to discover the method to utilize the scientific method, cell-free biochemistry, to discover how the body builds and metabolizes compounds. And everything that I told you about 20th century biochemistry, the story of cholesterol, but really the entire story of biochemistry, can be traced to his accidental discovery. Accidental discovery, okay? He found that a yeast extract that was made ruinously by accident was actually capable of bubbling up literally making bubbles that indicated that it was alive, but it wasn't. And so he taught us that you did not have to have a living organism to have the force of life. He taught us that life was chemistry. He taught us, therefore, that life can be understood as a chemical problem and therefore by the scientific method. And he left us with this wonderful statement from the early 1900s that we as scientists should never fall into the trap of thinking that there's a problem that we will never be able to understand, that we shall never know, and we should have confidence that whatever the problem is that we study, whatever the puzzle is, that someday it will be accessible to us as natural scientists, that there is no limit, in other words, to the power of the scientific method. I think that Buchner recognized that and practiced it in his day. I have tried in my lifetime to practice it in mine, and I hope that you will consider careers in science, and doing so understand that it's a calling of the highest order that has potentially the greatest impact of anything that we can do. So thank you very much for your attention. I'm sorry I can't be there with you today, but I hope that today's lecture has been of some value to you in thinking about what comes next for you.
Yeah, let's thank Professor Rothman for his very interesting lecture. Actually, as I told, he is going to visit the Academy of Sciences of Moldova, Republic of Moldova in October. And we will have the possibility to talk to him and to discuss these issues. Very interesting, the dependence he has shown the dependence of life expectancy as a function of time. And actually, durata vieții a început să crească odată cu dezvoltarea științei. Deci, și iată, într-o discuție prin Zoom, a spus că dacă nu era dezvoltată știința, durata media a vieții astăzi ar fi fost la nivelul între 25 și 30 de ani. Ladies and gentlemen, so we should go further. And the second lecture will be presented by Professor Klaus MAINZER, President of the European Academy of Sciences and Arts, Salzburg. And I would like to tell you that Professor MAINZER has been collaborating with the Academy of Sciences of Moldova for many years. On June 10, 2021, during the Science Week dedicated to the 60th anniversary of our Academy, he delivered the public lecture, Artificial Intelligence, Present Status and Future Prospects. On June 28, 2023, Professor Klaus MAINZER contributed to the success of the International Scientific Conference, Science and Art for Peace. A year later, on August 27th, 2024, on the Independence Day of the Republic of Moldova, Professor Klaus MAINZER delivered a lecture at the second edition of the summer school, The Path to Scientific Discoveries. For a long-standing, fruitful collaboration with the Academy of Sciences of Moldova, Professor Klaus MAINZER was awarded with the Dimitrie Cantemir Medal of the Academy of Sciences of Moldova. He visited us in December 2024. Professor Klaus MAINZER, are you online?
Yes. Can you see and hear me?
Yes, we see and hear you. Welcome.
Very good.
You have the floor.
At first, let me share my slide with you. I hope it will work. Okay. And now, Good. Now we can start.
Yeah, please.
Thank you very much again for this nice invitation to Moldova. You mentioned already I visited the wonderful hall where you are again. I recognize it. I have a very nice remembrance of that. And today the title of my talk is From Innovation to Application, Challenges of AI in Science and Healthcare. Because I thought the first speaker is a Nobel Prize winner for medicine, so I will consider especially applications in that field. Now, all that is on a long-term engagement in AI research, and these are books of mine which were published in the last years, and they document this engagement in that field. I will start on this background with the question, what can we learn from nature? And then I will consider innovation of AI in science, medicine, and healthcare. But then in the third part, I want to make clear that our classical digital computing and the classical digital AI is running into severe problems, into serious problems because of the energy consumption. And there I'm personally engaged in alternative computing, which is more brain orientated, and alternative AI, which is more brain orientated, and it is connected. That is called neuromorphic computing, and it is connected with quantum computing, where I'm also engaged, especially here in Munich, at my university, the Technical University of Munich. And in the end, I will come back to the question of responsibility of AI for the future. Now, let's start with the question, what can we learn from nature? To make a long story short, we can learn a lot. Especially, we can learn from the evolution, biological evolution of complex systems. That means in life science, you know, the atom of life is a cell. And a cell is already a very complex system of interacting molecules. And then on the next level, we can consider an organ in medicine. And an organ is a complex system of interacting cells. And on the next level, also still in medicine, an organism is a complex system of interacting organs. And then we can go on to a population in biology, which is a complex system of interacting animals, for example, organisms. And on the next level, finally, an ecological system, which is a complex system of interacting populations of animals, of plants, and so on. Now, from a formal point of view, a mathematical, physical point of view, all these systems are so-called open systems or dissipative systems. And that means that they are in a more or less permanent interaction with their environment by energetic, by matter, or by information interaction. And at certain critical values of this kind of interaction with the environment, we observe that more or less spontaneously, new order, new structures, new patterns emerge. And a prominent example is the brain. The brain is a complex system of billions of interacting cells, of neurons, and they are interacting by synaptic, electrical, neurochemical interactions. And here in this picture, you can see the brain with neural clusters. That means synchronously firing excited neurons, which are here illustrated by the colors yellow and red. That means pattern emergence, which I mentioned before, everywhere in nature and in complex system, now in the brain. And the amazing insight is that these patterns here in yellow and red, which are measured and observed by so-called PET measures, that means positron emission tomography measures, that these patterns are correlated with cognitive, with psychic states. For example, perceptions, emotions, thoughts, and consciousness. So a psychologist or brain researcher can see with these pictures what the individual patient is just doing. And now this activity of this complex system is illustrated here in a systematic way. On the left, on the macro level, you see the brain, and then on the micro level, the interaction of neurons and synapses, and here even in more detail. And when these neurons are excited, they fire, they generate an electrical signal which is sent to the neighbored cells. And that was at first time observed and measured in the 40s and the beginning 50s of the last century by two Nobel Prize winners in medicine that were Hodgkin and Huxley in Cambridge. And for the first time, they could measure these activity of the neurons. In that time, it was not possible to measure these very tiny synapses and neurons in the human brain. These were rather large neurons and synapses in octopus from the ocean. And by that, these physicians, by the way, they got a first mathematical model of the activity in the brain. So to say, a neurobiological model of the interactivities of the brain. And one of the rules they detected and wrote down in a mathematical equation was the rule that such a neuron is excited. That means it will fire an electrical signal if the inputs of the neighbored cells, the sum of the inputs of the neighbored cells, is larger than a certain threshold value. And therefore the sigma here, mathematically, you know, that is the symbol for a sum, summing up the neighboring inputs. Now, that was fine, and that inspired later on significantly the modern AI approach with machine learning, because in that approach, they used a simplified model of the human brain. The neural networks, in a simplified way, they are shown here. And the nodes here in this diagram represents neurons. The arrows represents the synaptic interaction. And in the model, these arrows are weighted by numbers. And the numbers indicate the intensity of the neurochemical intensity of the signals. And then, as I mentioned before, the rule now in the model, in this computational model, that such a node or a neuron is said to be excited if the sum of the neighbored inputs is larger than a certain threshold value. And by that, you get a process, you get a sequence of signals, a data stream, which is led through the whole architecture of the neural network. And you can see here the neural network is distinguished into certain layers, and that is also inspired by the neocortex of the human brain. The neocortex of the human brain, I think, has up to 6 layers. and that is more or less simulated here. Now, these data streams are triggered by learning algorithms. You know, the brain is learning by data, by experiments and sensory perceptions. And these kinds of learning algorithms nowadays are mentioned here. There is at first supervised learning. Supervised learning means at first we train the network by a certain prototype pattern that may be a photo of a face, and this face is represented by the distribution of pixels, millions of pixels, and they are trained with their different shadows and degrees of colors. They are trained to this artificial neural network by actually adapting the weights, which I mentioned before. That is done by commands in programs, computer language programs. Anyway, by that, the system is trained. It has a memory of this trained photo, and then it is able to detect this pre-trained photo among a huge number of other perceived faces or photos by comparison in detail the pixels in that way. Now, that was supervised learning. Non-supervised learning means that a system, such a neural network, is more or less spontaneously able to detect similarities of data, and then according to this criteria, it is able to classify a whole class or mass of data. Reinforcement learning means that the system is able to spontaneously improve a certain solution procedure by rewards from outside. It's like a child. A child in a classroom gets rewards or critics by a teacher, and then it is able to improve its behavior or its answers. And later on, we will see that these spectacular chatbots today are actually acting with this kind of algorithm to improve its answers. Now, deep learning only relates to the depth of the neural network. As I mentioned, there are several layers, and depth means the number of layers. Now, in the history of AI, at first, a very simple neural network was considered with only one layer of neurons. And now an interesting aspect. Modern machine learning is not only inspired by brain research and the human brain, but also by physics, because the first AI researchers with real technical breakthroughs in that field were no physicians, no biologists, no computer scientists. They were physicists, and they were inspired by materials, physical materials. John Hopfield, for example, was actually a solid-state physicist. And what do solid-state physicists? They analyze solid bodies. For example, here in the picture, a ferromagnet or spin glasses. A ferromagnet is again a complex system, which I mentioned before. A complex system which consists of many micro elements. In this case, they are atomic dipoles. Atomic dipoles means with two states, which are illustrated here by the arrow pointing up and down. And now this is a more or less random distribution of these microstates of the atomic dipoles in this solid body, a ferromagnet, and then the system is annealed to a certain critical degree of temperature. In this case, it is a Curie point. And then we can observe on the micro level that these dipoles are more or less jumping into one direction. And that means that the whole state is self-enforcing. And so the whole system became magnetized. And that means that from this order distribution, a new regular order emerged by this kind of phase transition. Now that was physics. And the idea of Hopfield was to use the mathematics here, which describes this process, to transform it into a cognitive system. For that, instead of the atomic dipoles, now nodes are considered. Technical neural cells with two states instead of the alternative dipole direction, now two states, black and white. And this kind of irregular distribution is illustrated here. And then instead of the phase transition in physics, that means annealing to a temperature, now the mathematics is interpreted as a learning algorithm with which we can approximate a pre-trained regular pattern. For example, the regular pattern here of the shape of the letter A. So again, order emerge from disorder now by the learning algorithm, and that can be interpreted as a recognition process of a system for a certain order. And for that, Hopfield got the Nobel Prize in the last year in physics, because he is a trained physicist, and it underlines the deep rooting of modern AI in physics, of course, also in mathematics. And that was generalized in the next years, such as in the human brain, not only one layer, but several layers. You remember the neocortex of the human brain? That is illustrated here on the left, only for 4 layers. That means on the bottom here, there are only colored pixels which are distinguished, and then on the next level they are connected as edges and corners, and then next level partial faces, and then the whole faces. And actually that's all. That is modern machine learning. The great difference is in the brain, I mentioned it already, we have 6 layers, but in the real world, artificial intelligence network nowadays, we have not only 100, we have thousands of layers, we have billions of neurons and technical synapses, and therefore this amazing results which we can observe nowadays. But in principle, here the basics are explained, and the application and development for multi-layered neural networks were mainly done by Geoffrey Hinton, and therefore Hinton also got the Nobel Prize for physics as AI researcher in the last year. Now, these are the foundations. Let us consider now applications. And we start again with physics. And here, a very nice, I think, exciting example. 2018, CERN, you know CERN, the Center of Nuclear and the Center of Elementary Particle High Energy Research in Switzerland, they came up with the announcement that now the Higgs particle is detected. The Higgs particle, by the way, is a prominent particle in elementary particle physics, which can explain the emergence of mass in the universe. So it is very crucial. And now what had happened? It was detected by supervised learning. You remember, supervised learning means at first there must be a prototype, exactly a supervision. That was done by Peter Higgs. the later Nobel Prize winner. And Peter Higgs, as a theoretical physicist, he predicted mathematically by deriving from the axioms of elementary particle physics theory. He derived mathematically the existence of such a particle, which was later on named after him, the Higgs particle. And he predicted even more, namely that this particle would decay into a certain pattern of other particles. So he predicted mathematically a certain pattern, we can say a fingerprint for the existence of the Higgs particle, a certain pattern. Now supervised learning means the system, the AI system, is trained by this fingerprint, by this pattern, and then you remember the recognition of faces by these systems, and then it was able to detect in a huge mass of data. And these data are generated by the collision in the accelerators in CERN, by the collision of accelerated protons, and billions and billions of data, and it was able to detect this fingerprint, which is illustrated here in red. These are these particles which are connected with the emergence of the Higgs particle here in red, and all the other particles which are generated and data after the proton collision are here in blue. Now, the amazing fact is that the same kind of process, the same kind of AI algorithm is applied in a completely different field, namely in medicine. For example, here in breast cancer, at first, supervised learning, the system is trained with the criteria of cancer cells. And then it is able to separate the distribution of cancer cell in the breast, in breast cancer, here in red, from the other healthy lymph nodes. This is another example, machine learning in biochemistry, in medicine too, AlphaFold, which also came up in 2018. It is a multi-layered neural network which can discover the shapes and foldings of proteins. You know, proteins are crucial in a cell for all kinds of life functions, and they are determined in their reactions and behavior by a certain shape, the folding of the protein. And that is a standard folding. And if there are tiny deviations, they may be a hint for the emergence, for example, of cancer tumors and so on. So what here the algorithm is doing, it is analyzing a protein as a sequence of amino acids. The amino acids are represented here by these Latin letters, and then tiny perturbations in the normed sequence of the folding is detected by this kind of algorithm, and that may, as I already mentioned, be a hint for the early detection of emergence of cancer tumors or other diseases. Now we can go on and with this kind of machine learning algorithm. It is nowadays responsible for the enormous acceleration of the production of new medicaments. Actually, in former times, the design and production of new medicaments needs a time scaling which we know from engineering. For example, engineering an aeroplane, years and years, but now it is significantly shortened in time. And this algorithm can also be applied to detect new correlations, for example, which were never before recognized by human scientists, correlations between between a chemical substance and, for example, a cancer tumor. This kind of reaction were detected here by AI algorithm. Now, applications also in medicine, in neuro robotics. Here, chips with analog components are able to predict and to prevent seizures. That may be Parkinson and epilepsy diseases. And here in the picture, it is illustrated, number one. That means there is a procedure with a chip which is able to detect changes in the states. That means it can be used as an early warning system for upcoming seizures. And then two, illustrates how now reactive treatment can be initiated in order to prevent the seizure. And another example on that line in with neuroprothesis in neuro robotics, the interaction of humans, human organism tissues, nervous tissues with the technical prosthesis, for example, that is illustrated here. Learning neural net encoders transfer sensor or motor steering signals into parallel sequences of pulses for regulated, implemented microcontacts here in nervous tissues. Now, in medicine, the AI tools are not only applied here in high-tech surgery, but they especially nowadays are necessary for the infrastructure of medical health care. And that is so important that became obvious in the time of the pandemics. You know, I remember in Germany, they used high-tech in the surgery, but the infrastructure was very poor. And so this is illustrated here in the diagram. Infrastructure means the digital interaction between patients, medical carer, service provider, and so on, to have an effective interaction for the health of the patients. Now, all that is fine. All that is fine nowadays with digital tools and AI tools. But now I come to a crucial aspect, namely the digital equipment nowadays is based on digital classical computers, and with that we will run in the next year in severe problems. And therefore I'm personally engaged here in more brain-oriented computing and finally quantum AI, and that would now be the next part. As I mentioned, all these nice applications which I mentioned before are realized on digital computers. These are the so-called von Neumann computers. The architecture is still von Neumann. You know, von Neumann technically realized the first digital program-controlled electronic computer already in the 40s, but the architecture is still the same even on our digital chips nowadays, in your smartphones, in our classical supercomputers. And the von Neumann architecture means, here on the left, illustrated here, there is a strict separation between the memory unit and the processor unit. And that is even true for the so-called graphic processor units, not only the classical central processor units. The strict separation between memory and a processor. And that means that with the increasing mass of data nowadays, all these data must be pressed through this architecture. And that needs a lot of energy. It needs a lot of time. So in some years, the major part of energy consumption on this earth will be in digitalization with this kind of architecture. Now we can learn again from nature. In nature, energy efficiency is amazing. Our brain only needs 20 watts, 20 to 25 watts. Amazing for all these nice abilities. And the main reason is that here is a completely different computational architecture. No longer the strict separation of memory and test, but a network of neurons with a synapses. And in the living brain, the living synapses, they can integrate both. No separation. Here, processor, their memory, the synapse, living synapse can both processing and memory. And there is not only one synapse, there are billions of them in the brain. So what we nowadays call the von Neumann bottleneck, by this von Neumann architecture, that is no danger in the living brain. But these networks, you know, are already used in machine learning, but they are only simulated. They are simulated on classical digital computers, and they need still an enormous mass of energy. So the idea came up, can we realize these networks in hardware, such as in the brain, in redware? Is that possible? The answer is yes, we can. And first, the idea came up in the 70s, the 80s of the last century by an a professor of electrical engineering in UC Berkeley. And he theoretically suggested a so-called memristor. That means a kind of transistor which is able to integrate, like the living synapses, the processor and the memory function. And that is here mentioned for technology. The memristor, is it called? Memristor is a combination of memory and resistor. And that integrates processor and memory. And that is theoretically described here, technically described here in a diagram. And I wrote with Chua a lot of books on the mathematics, me more on the mathematical, theoretical side. He is the electrical engineer. And that is illustrated here, a layer. You remember the Hopfield layer. And the synaptic interaction is now realized by a memristor. So we have the Hodgkin-Huxley level. That means the mathematical description of the neurobiological interaction of the brain, which I mentioned before. And now the electrotechnical model with the memristor, which is mimicking the single synapses. Now, that is fine. The mathematics is wonderful. And every professor of electrical engineering and mathematician will like that. But the great question was, can we do that in reality? Physically, yes, we can. The point is, we must find materials, smart materials, with a kind of memory. And an example is, Intel, the Silicon Valley company in the beginning of this century, came first up with titanium dioxide. That is a material, if you apply charge to it, then the molecular structure is changing. And by that, this material has a kind of memory of a former state. And that was used to realize it. Here, the lecture is mainly for pupils, I heard, at the grammar schools. And we all learn at the grammar school, I hope so, we learned all Ohm's law. Ohm's law means voltage is proportional, that is here, V, voltage is proportional to current, I. And the proportional factor, R, means resistance. But in the classical version, that is a linear equation, and that means R is a constant. And if you illustrate that here in the diagram, voltage and current, then in the classical case of Ohm's law, you will get a straight line because it is constant. Now here I mentioned permanently charge is applied to the material. That means the state of the material is not constant, but it is changing by charge. Mathematically, resistance depends on charge. And so, mathematically speaking, the equation is no longer linear, it is a nonlinear equation. And that is illustrated here. You get in this case a kind of curve, nonlinear, and this so-called curve of hysteresis is a signature for all kind of these smart materials which have this memory capacity. And that is amazing. Nowadays, a lot of new materials were detected which have this kind of signature in the behavior. They can be used as synapses, technical synapses in neural networks. You see here one layer of this network, again with the synapses here mimicked by the memristus, here again illustrated. And that is actually amazing. And with this electrotechnical model, we came closer and closer to the brain-oriented activities which I described before. Now, I'm on the background of this research, I'm now in the advisory board of the German Ministry of Research for the so-called neuromorphic systems AI. That means brain-oriented systems. And they are in these future clusters of research, they are not only modeling it mathematically and technically models, but they want to build it, and they do it together with the industry with this kind of materials. Now, being in these research clusters is very exciting, because some years ago a new idea came up, namely, instead of electrons in an electronic network, let us consider photonic networks. Instead of electrons, photons in laser light. And it is no longer between electronical units mimicking the neurons, but here the light between optoelectronical units mimicking these optical neurons. And I cannot go here by reasons of time in the details. What we get is a kind of brain of light, I think I want to call that. And I like it. It is still in an infancy, this kind of research, but we already know it will have enormous improvement considering with the memoristic and electronic approach, which I explained before. Namely, instead of the milliseconds of electrons, it now only needs nanoseconds of photons. And concerning the energy consumption, it is far more energetically efficient, nearly to 75 watt. And that is amazing. You remember the brain needs 20 to 25 watt. That means with the photonic approach, we come very, very close to the efficiency of the human biological brain. Now, anyway, what I also want to highlight here is the photonic approach. Of course, it is a future perspective, also can be connected with another prominent application of computing nowadays, and that is quantum computing. And here in Munich, I'm also engaged since several years in that field. There's a book of mine of quantum computing, and I only want to mention here some highlights which are typical for quantum computing. The main difference to classical computing is the application of so-called quantum bits. Not only bits, but quantum bits. What does it mean? Actually, very simply to understand. Classical bits, you know, either or, alternative states, 1 or 0. And all kind of information can be represented by this kind of sequences. That is classical digital computing. But here, the case of quantum computing, we also can consider the intermediate states between 0 and 1, namely in the sense to be more probable closer to 0 and more probable to be 1. And so it's a continuous scaling which is packed together in a so-called quantum bit. Now that is mathematically described here. I will not explain the symbolic here. That is the typical language of quantum mechanics, but the idea of a quantum bit, I think, is very clear. Now, concerning the hardware we want to build that, we can use photons. That is a link to this photonic neuromorphic brain, which I mentioned before. We can use a photon, and a photon at first can be used to realize the classical bits, 0 and 1, by polarization. You all know in summertime the polarized sunglasses, and here that means that such a photon can be vertically or horizontally polarized. And that represents 0 and 1. And then in the sense which I described before, we can consider the intermediate state between 0 and 1, and actually the quantum bit is realized in the quantum system of a photon. The point is nowadays, if you look in the labs of quantum computing, they are not only realized by photons, there are a lot of other materials which are used, for example, electrons, atoms, ions, or quantum dots, and so on. And it is a kind of race or competition in research, and we will see who will win the competition in the end for building a quantum computer in hardware. My personal impression is, in the end, the photonic approach will have the most advantages, but we will see in future. Now, the main feature, one of the main principles in quantum mechanics, which explains the enormous advantage of quantum computing considered with the classical approach, is the so-called property of superposition. Superposition I explained already with the quantum bit. That means that contrary to classical computing with the von Neumann computer, you must not elaborate in a sequence of bits, each bit after the other. And that needs the enormous energy. Each bit must be stored and things like that. But here in the quantum computer, all these bits, millions of bits, can be put into one quantum state. They are connected in one state. That is called a superposition of these states. And then a computational device is applied to the whole superposition of all these single bits, not bit after one another. And by that, you get an enormous acceleration of computation velocity, of course, and by that it is explainable that the problems which could not be solved in human life can now be solved in perhaps minutes or seconds, and that was already mathematically at least proof exactly in the end of the last century for the so-called factorization problem with the Shaw algorithm and so on. The mathematics and the theory is fine already, but we are now in the process of realizing it technically. And besides the superposition, I want to highlight another main property of quantum mechanics, and that is the so-called entanglement. Actually, entanglement was a philosophical question. And who came up with this deep idea? That was Einstein. Einstein in 1935, he analyzed the formalism and of quantum mechanics, and he said, amazing, we here have to do with a new kind of reality, namely, which is not understandable in classical physics, distant locations, far away particles at far away locations, they are actually in a common state, independent of the distance of these two particles, for example. And that was later on confirmed experimentally by spre in the beginning of the 80s. And nowadays, it is the basic in technology, revolutionary technology, namely quantum communication. That means, and that is illustrated here on the left in technology, which is nowadays already used in China. We have far distant locations here, Earth stations, and then they are correlated, they are entangled in this common state by a satellite. From the satellite here, photonic rays are sent to the different stations, and then these two photon rays are entangled. They are prepared in a common state. And the result is that the perturbation of these networks, you can imagine not only two, but several, a network of these stations, like in a quantum internet, and a local perturbation here is immediately perceived at every location in this network. So in the end, the great advantage of such a kind of quantum internet will be, in a technical sense, absolute security. That means you can detect immediately the perturbations or a cyber attack or whatsoever. So I'm convinced that will be the future of the internet in some years. Now let me come to the end with some remarks towards innovative and responsible AI. Some of you will already waiting for this application of AI, because that is the application. Everyone knows that nowadays. That is ChatGPT. These are, with these kind of chatbots, AI has arrived from the labs in research in our everyday world. Everyone knows ChatGPT, only 1.5 years old. And that means it is a chatbot on the basis of large language models. That means a huge mass of data with algorithm is producing these amazing simulation of human intelligence. And by the way, ChatGPT, that is of course US, that is open AI, one of the most wealthy startups in the world with billions of dollars. And but it has a competitor nowadays, and that is China. 1 year ago, China came up with DeepSeek, and by the way, DeepSeek is needs only a minimal of the energy of ChatGPT. It is much more efficient energetically, and it does not need these very expensive AI chips of the US product. But anyway, both are fine approaches here, and people are amazed, amused. Some people are horrified by this kind of simulation. We all know that. But let me say, my point of view, a mathematical point of view, don't worry, because let us have a glance in the machine room of these chatbots. And that's very simple, because it is working with two well-known algorithms, which I explained before. First of all, supervised learning. Supervised learning, you remember, means that at first the system must be trained by a mass of data. And then the second algorithm is much more interesting. That is reinforcement learning. And we all know that reinforcement learning, because if you interact with ChatGPT, for example, then you know if you are not satisfied with the answer and you criticize ChatGPT, then it is able more or less to improve its answer and to learn again of this kind of interaction. But behind that, from a mathematical point of view, there is nothing else than statistics. That means in the large language models, they are computations of statistical expectation values of future words or letters which should be inserted here in a certain mask of a pattern of words. Nothing else. And therefore, I like to call these systems a stochastic or a statistic parrot. But anyway, some AI researchers are already speculating that we are on the way to an artificial general intelligence with the abbreviations AGI. And is that true? I am here very reserved, because on this line, we will not realize the artificial general intelligence. But what we can do, amazing. We can do a lot of statistical approaches to simulate human intelligence and even to overcome the possibilities of human problem solving. The example here is that ChatGPT is only reserved to large language models. That means it is mimicking language abilities. But you know, human intelligence is by no means only speaking and communicating. Human intelligence is acting. Human intelligence is perceiving. Human intelligence is detecting by pattern recognition. And that is now on the way with so-called robot GPT, not ChatGPT. Robot GPT means that we do not only have the large language models, but we also have large visual models, we have large sensor models, and we have large motor models. And by that, we can build robots on this basis, such as the ChatGPT. That is illustrated here. If we have a glance in the machine room, that it is quite similar to the ChatGPT, but it is extended, the language, large language models, by mass of data, of sensory data, of visual data, of motor data. And by this mass of data, the system can learn, here the robot can learn such like an human being or such like an animal. But that is only one strategy here in technology and AI, and by no means all possibilities of the brain are explained by that. But it will be rather successful and a nice tool for the future. In the end, I want also to underline, some people believe intelligence means that there is a human-like entity, technical entity like a robot, which is much more intelligent than humans. That is a typical idea and vision in science fiction. No, which is even more amazing nowadays is that there are impersonal infrastructures, and they are intelligent in the described way. And that is illustrated here. That is really the future here on our planet Earth, that these kind of infrastructures, digital intelligent infrastructures, are growing together, like here, this brain that is, of course, a metaphor, which is spreading all over the planet Earth. And here on the right and left, you find the highlights of infrastructure which are already exist: communication, global communication networks, or circular economy, for example. That means the idea, in order to avoid waste and pollution, that the economy is self-regulating in some sense, like an organism, like a system, in the way that it is able to avoid pollution and waste by incorporating the residuals of production in the production chain, and by that it is realizing what we nowadays call a circular economy with feedback loops, or the global financial systems, which would be not possible without these kind of fast AI algorithm. And the health system infrastructure I already explained. And last but not least, we are living in times of wars. And that means that instead of the battles with tanks in the 20th century, we now have the battles of more or less autonomous drones, for example, which are made possible by AI equipment. And at least the last example also illustrate us in a drastic way the problematic aspects of this kind of revolutionary technology. That is my last slide. I come back to the origin of science here in Europe. You remember that was done in the time of Greek antiquity, and Hippocrates, especially in medicine, one of the first who established what we nowadays would call an evidence or empirically based medicine by observation. But it is important. That is typical for the origin of medicine at that time, that this kind of scientific insight was combined with an ethical attitude of physician, the famous Hippocrates oath, which requested human well-being as measure of healing and helping. And in that sense, I think it is our challenge for the future that these AI tools, which I mentioned before, also should be used as innovative service system for mankind to handle the increasing complexity in science, health, and civilization. I thank you for your attention.
Dear Professor MAINZER, thank you very much for the wonderful lecture. I would like to note that your lecture demonstrated that science is fascinating, and nowadays science is multidisciplinary. You gave so many examples where it's interpenetration of different disciplines. You spoke about mathematics, physics, medicine, also made references to electrons, to photons, to photonic, optoelectronic structure, and so on. Well, we actually do not have time to open the session for questions, answers. I know that you should get a train. I know that. But I would like to know that I got already via mail some, how to say, that means people from the Republic of Moldova, also from Romania, I got some messages, and even a message from Japan. That means your lecture has been live streamed for our society, but live streaming, it means that everybody on our small world can had the possibility to follow your lecture. It was really a wonderful lecture. Thank you so much. The lecture, actually, I tell to our pupils, the lecture will be available on the portal of the Academy of Sciences of Moldova. And well, you can enjoy it again and again, and especially other pupils, students that had no possibility today to follow, they will have the possibility to. So, dear Professor MAINZER, are you still online? He probably, da, eu o să trec atunci la limba. So, are you online still?
Yes, now you know
Yeah.
I must rush to the train to the next
So,
event.
thank you.
Yeah.
Thank you so much. And well, we actually will wait for you in Moldova. Again, we will have a conference devoted to impact of artificial intelligence on different fields of human activities. And well, you will be very, very welcome, or at least to give a lecture online. Thank you so much.
Yes, and I look forward to come again to your wonderful country. Thank you so much. To enjoy the wonderful grapes and wine.
Thank you.
Yeah,
Thank you.
good. Thank you.
So, all the best to you. Ladies and gentlemen, we come back to the Blue Hole. Mai avem o lecție, o prelegere astăzi și domnul academician Aurelian GULEA deci va prezenta perspectivele de tratament al cancerului. Deci iarăși este o prelegere multidisciplinară. Domnul academician deci activează în domeniul chimiei, așa ca bază, dar deja de mulți ani realizează proiecte la intersecție cu medicina. Și astăzi noi avem posibilitatea să savurăm rezultatele care au fost obținute, spunem așa, la intersecția chimiei cu medicina. Domnul Câncian, you have the floor.
Mulțumesc, domnule președinte. Astăzi noi, într-adevăr, am fost martori la niște lecții foarte importante și foarte interesante, deci legate de diferite domenii, dar fir roșu în aceste lecții a fost științele fundamentale. Și la primul raport și la al doilea, adică științele fundamentale, să are vedere și matematica, și fizica, și chimia, și alte care stau la baza celorlalte cercetări în medicină, după cum vedeți, aplicații în medicină. Eu sunt de formațiune chimist, cum a spus domnul președinte, și voi încerca prin prisma chimistului, deci să aduc la zi acele realizări care sunt astăzi în tratarea unei maladii foarte importante în viața noastră, care este pe locul 2, după bolile cardiovasculare. Și chimia este una din acele științe care încearcă, printre primele, să influențeze dezvoltarea acelor celule rele sau celule de cancer care se dezvoltă în organismul nostru. Eu vă salut pe voi, bună ziua și noi ne-am mai întâlnit și anul trecut, nu știu cine a mai fost, dacă mai sunt din ceea. Cu mare plăcere vreau să vă contactez. Îi spuneam, domnule președinte, că o lună în urmă am avut o întâlnire și cu elevii din clasa a 4-a. Deci, care erau tot interesați de anumite cercetări din domeniul chimiei, dar nu anume legate de cancer, dar deci așa ca curiozitate, dar mai mult îl interesa medaliile pe care le aveam eu cu mine, deci decât să zicem subiectul legat științific. Eu cred că cu voi vom găsi o limbă mai apropiată, mai comună, deci ca să putem comunica în privința aceasta, fiindcă ceea ce voi încerca eu astăzi să vă spun este de fapt legat foarte mult de sănătatea noastră. Adică încercăm să cunoaștem care sunt pricinile și care pot fi metodele de tratament ale cancerului din punct de vedere al științei moderne. Dacă să luăm, deci, pe bune, ce provoacă cancerul, asta în primul rând sunt energiile nefaste, radioactivități, adică atunci când în organismul nostru se influențează din exterior cu energii foarte mari. Astfel se formează radicali care nu sunt controlați. Acești radicali, mai departe, pot să influențeze asupra celulelor noastre, provocând mutații sau, mai bine zis, se formează celule necontrolate sau celule bolnave, celule de cancer. Noi, ca chimiști, încercăm să construim molecule care să poată stăvili, adică să oprească dezvoltarea acestor celule care au acțiune nefastă asupra organismului nostru. Deci, acel prim pas pe care îl fac chimiștii este de a construi niște molecule. Molecule apte să fie cu capacități anticancer. Eu vin tot din știința fundamentală, adică până la vârsta de vreo 50 de ani, eu m-am ocupat cu știință fundamentală. Este vorba de rezonanța magnetică nucleară. Deci, o metodă modernă care, în principiu, dă posibilitatea să studiezi proprietățile la substanțe chimice, studiind nucleul atomului, adică proprietățile chimice prin proprietățile nucleului. Este o direcție, să zicem, foarte interesantă și foarte importantă, mai ales când e vorba de medicină, fiindcă noi putem să studiem proprietățile acestea chimice, având la dispoziție niște cercetări fundamentale foarte importante. Spre exemplu, remenia, adică rezonanța magnetică nucleară, se face pe protoni sau carbon 13, unde sunt nuclee magnetice. Un nucleu magnetic poate să interacționeze cu un câmp magnetic, având posibilitatea de a rezona, de a da un fenomen de rezonanță. Astfel, noi putem studia aceste proprietăți chimice cu această rezonanță magnetică nucleară. Prin ce ea este deosebită? Iată, luăm molecula de apă. Molecula de apă simplă, da? Care este formula moleculei de apă? Știu toți, da? Este formată dintr-un atom de oxigen și 2 atomi de hidrogen, așa? Dar H2O, cum spunem noi, asta poate exista în ce condiții? Iată, dacă avem un pahar cu apă sau apa care este aici, molecula de apă nu este H2O, dar este la temperatura de cameră, este H8O4. Dacă o să ridicăm temperatura, putem ajunge și la formula care a spus noi H2O, când este starea de vapori. Sau dacă ne coborăm mai jos de temperatura de cameră, putem să avem altă formulă a apei. Vedeți că nu e chiar așa de simplu, nu este numai H2O. Iată, rezonanța magnetică nucleară poate să studieze aceste fenomene și să ne dea această informație. Dar cel mai important este remenede informația despre faptul cum se mișcă moleculele în sistemul respectiv. Sau mai bine zis, timpul de viață. Timpul în care o moleculă de apă își schimbă poziția egală cu dimensiunea ei, care e de fapt de ordinul de 1.000.000 de secundă. Dar fiindcă organismele umane sunt formate din apă mai mult, deci e clar că este important să studiem acest fenomen, adică ce se întâmplă cu apă în organismul viu. Și pe urmă acesta ne dă posibilitatea ca mai departe noi să putem construi molecule care să fie de folos, active, care să poată combate celulele nefaste. Deci asta este pentru noi o lecție importantă de a ști care este deci, structura substanțelor în mediul apus, pentru că noi, ca om, ca substanță, ca materie biologică, deci avem această proprietate de a conviețui într-un sistem apus, într-un sistem în care moleculele de apă joacă un rol important. Și ce vă spuneam eu despre molecule că se formează H2O de 2 ori, sau de 4 ori, sau de 8 ori, asta este legat prin faptul că între aceste molecule se formează legăturile de hidrogen. Legături de hidrogen foarte labile. Și de aici ajungem și la cel mai important, la celulă. În celulă avem nucleul, ADN-ul, care în principiu tot la bază are formarea legăturilor de hidrogen. Și prin urmare RMN aici devine un instrument foarte important de studiu a comportamentului atât a apei cât și a moleculelor pe care noi le construim. Și această activitate a lor poate fi previzibilă, adică noi o putem într-un fel programa, așa, ca să știm care molecule și cum acționează prin intermediul acesta. La baza cercetărilor care au urmat după cercetările mele legate de știința exactă, deci știința fundamentală, a fost o deplasare a mea în Canada, pe o bursă de la Guvernul Canadei, unde am făcut o stagiune de 4 luni într-un centru oncologic, unde majoritatea chimiștilor făceau molecule, construiau molecule care să fie cu proprietăți anticancer. Și acest laborator de STIT, acolo au fost descoperiți, spre exemplu, o analiză la cancer la prostată. Profesorul Labrie. Deci, metodă care se folosește în toată lumea. PSA se numește altfel, deci analiza aceasta este importantă pentru bărbați mai mult, deci pentru că e vorba de prostată. Iar tot acolo, chimiștii au dezvoltat și medicamente care astăzi, dacă este depistat cancerul la prostată, este lecuit. Deci sunt remedii, medicamente care pot să lecuiască cancerul prostatei. Deci acolo, având și eu ca chimist un bagaj de substanțe de la Chișinău, am încercat să fac un screening la acele substanțe pe care chimiștii moldoveni le-au sintetizat în laboratoarele de la Universitatea de Stat, la Institutul de Chimie. Aceste prime încercări au fost încurajatoare, fiindcă am găsit unele din substanțe care, într-adevăr, au o activitate bună, adică omoară celule de cancer. Ce să vă spun? E clar că astăzi, dacă să ne uităm prin prisma ce este important în tratarea cancerului, eu aș spune, sunt 3 lucruri. Primul, trebuie să avem molecule care sunt active, foarte active. Adică în cantități minime se poate omorî celule de cancer. Ceea ce este astăzi în medicină, asta e 10 la minus 6 mol pe litru, adică e vorba de grame. Când se tratează această maladie, omul este medicamentos tratat cu grame de substanță, cum ar fi cisplatina sau doxorubicina și altele. Ceea ce am reușit noi să avem, deci substanțe foarte active, înseamnă că putem trata cu miligrame, adică de mii de ori. Doza este mai mică, dar noi știm că cei care trec tratamentul cu deci, chimie sau chimioterapie îl trec foarte greu. De ce? Fiindcă dozele sunt foarte mari. Deci dacă noi putem diminua aceste doze, înseamnă că pacientul poate fi tratat în condiții mai blânde. Acesta este un atu. A doilea, deci, este vorba ca moleculele pe care noi le construim să fie selective, adică să atace celule de cancer și să lăse în pace cele sănătoase. Ceea ce avem noi astăzi în tratament, de obicei, cisplatina sau doxorubicina, omoară în același mod sau în aceeași cantitate și celulele de cancer și celulele sănătoase. Deci, o problemă este selectivitatea, de a construi astfel de molecule care selectiv să acționeze asupra celulelor, lăsând în pace cele normale și lichidând cele care sunt bolnave. Și nu mai puțin important este și următorul fapt. Deci tot o problemă este ca substanțele pe care noi le facem sau molecule pe care noi le facem să fie puțin toxice. Să nu fie toxice. Deci iată 3 piloni asupra cărora noi trebuie să adresăm deci, cunoștințele noastre pentru a construi molecule deci, de acest gen, de nouă generație, pentru tratarea individuală. Adică un bolnav poate fi tratat individual, fiindcă având același cancer la fiecare persoană este diferit tratată. Fiindcă noi fiecare suntem un individ, adică suntem toți foarte individuali, nu suntem toți tot una. De aceea, pentru a trata astăzi maladia de cancer, trebuie să fie, deci acest principiu, tratarea la nivel individual. Adică ce înseamnă aceasta? De la fiecare bolnav se ia celulele de cancer, se plantează și se testează pe diferite forme de substanțe anticancer și se alege anume aceea care este mai activă, care este mai convenabilă pentru persoana dată. Vorbim de medicină personalizată. Acum, dacă să trecem la concret ce noi am reușit să facem, e clar că primul medicament care îl cunoașteți cu toții a fost substanța chimică cisplatina. Este compus care este destul de activ. Este activ la concentrații destul de mari, 10 la minus 6 mol pe litru, dar deci are neajunsuri că sunt multe efecte secundare la tratare cu acesta. La ce trebuie de atras atenție? Este vorba despre substanțe care acționează asupra ADN-ului. Adică tratarea cancerului are succes dacă vom face astfel de molecule care direct sau intim să interacționeze cu ADN-ul pentru ca să oprească dezvoltarea acestora. Dar, deoarece ADN-ul se află în nucleul celulei, clar că pentru ca substanța noastră să penetreze în interiorul acestei celule, trebuie să treacă membrana. Dar membrana este cea care protejează. Deci substanțele pe care noi le construim trebuie să fie apte să treacă această barieră, să conțină așa fel de radicali, mai bine zis, deci nu radicali în sensul electronilor, dar radicali chimici care să penetreze în această moleculă astfel ca să ajungă la nucleu. Deci protejarea este făcută special în în felul următor ca să dea voie la acele substanțe care se dizolvă în grăsimi, lipide. Ajungând la ADN-ul. Deci, în ADN-ul am spus că un rol important îl joacă legăturile de hidrogen între nucleele de citozină, tiamidă sau adenină și guanină. Iată, aceste legături care sunt de hidrogen, ele sunt slabe, dar ele duc la formarea ADN-ului. Deci noi ar trebui să facem astfel de substanță care să penetreze aici și să înlocuiască molecule de adenină sau de citozină. Astfel, dacă în componența ADN-ului apare o moleculă care nu este recunoscută, atunci celula moare. Adică noi încercăm din interior să intrăm în această în ADN-ul. Iată, deci aici este un exemplu în care avem în componența ADN-ului, deci fragment care poate să formeze legăturile de hidrogen și aceste legături de hidrogen, dacă din punct de vedere energetic vom calcula, vom vedea că ele sunt foarte slabe, undeva de 2, 3, 5 kJ pe mol. Deci energii foarte mici și labile. Adică noi putem să intervenim aici cu moleculele noastre. Și atunci, iată un exemplu. O semicarbazonă care poate să interacționeze cu ADN-ul și, într-adevăr, legăturile de hidrogen variază în dependență de natura lor. Azot, hidrogen, oxigen, 8 kJ sau azot, hidrogen, azot, puțin mai puternici. Însă legături puternice de hidrogen nu sunt caracteristice pentru a forma medicamente. Deci astfel putem constata că ADN-ul este principala țintă în tratamentul acestor boli. Deci agenții care noi putem recomanda sau care pe de-a lungul anilor au fost sintetizați sunt agenți de alchilare, agenți intercalare, inhibitori de transcripție a ADN-ului și agenți antimetaboliți de interacțiune cu enzime. Aici avem o parte din aceste substanțe anticancer. Vreau să atrag mai mult la ultima, la cisplatina. Deci ea a fost prima substanță chimică care a fost utilizată la tratarea cancerului. De ce înseamnă cisplatina? Avem 2 atomi de clor de o parte, adică și celelalte 2 molecule de amoniac și cealaltă, adică sunt alături. Cis înseamnă alături, trans înseamnă vis-a-vis. Și prima concluzie care s-a făcut de interacțiune a fost că această moleculă, datorită geometriei sale, este activă, fiindcă dacă luăm și schimbăm poziția atomului de clor și amoniac și vine un compozit cum ar fi nu cis, dar transplatină, ea are activitate zero. Dar această concluzie a fost de fapt greșită, fiindcă adevărat mecanism care a fost aici demonstrat este că această cisplatină în mediul apos, clorul este înlocuit cu apă, iar această poziție fiind foarte labilă, este înlocuită de atomul de azot în poziția 7 de la guanină și astfel cisplatina intră în interiorul ADN-ului, iar când are loc replica, această celulă moare, fiindcă cisplatina nu este recunoscută. Deci care este totuși mecanismul de acțiune a cisplatinei? Nu este cis-trans, este acțiunul cinetic, adică timpul de viață a acestei molecule în apă. Vedeți? Clorul este înlocuit de apă și timpul de înlocuire este egal aproximativ cu o secundă. Iar timpul de replică a ADN-ului este egal tot aproximativ cu o secundă. Adică în acest timp are loc penetrarea deci cisplatinei, care se unește cu azotul de la guanină și astfel are loc înlocuire și mai departe, când are loc translare, se vede că n-a luat ceea ce trebuie. Și atunci celula moare. Astfel, deci, are loc celula de cancer este distrusă. Alți agenți sunt cei de intercalare, adică sunt molecule foarte mari care în compoziția lor au funcții sau grupe funcționale capabile să formeze legături de hidrogen cu ADN-ul. Iată cum ar fi doxorubicina în cazul de față. Deci, în interiorul ADN-ului, deci această interacțiune cu molecule intercalare duce la faptul că se formează compuși intermediari care mai departe duc la distrugerea celulelor de cancer. Și al treilea tip de agenți anticancer sunt cei care formează legături cu enzimele. De fapt, este vorba de metale. Dacă noi introducem în afară de aceste substanțe organice și metale pot lega ADN-ul prin faptul că enzimele importante sunt legate de către aceste metale. Un exemplu foarte important aici sunt compuși metalici care se formează cu enzime, care au fost sintetizate la noi în laborator, unde s-a demonstrat structura prin raze X și încă o paranteză de a se facă primele cercetări cu substanțe organice au fost efectuate în laboratorul din Canada, la Universitatea Laval, unde ca substanțe active au fost substanțele steroide, cu nuclee steroidice și piperazină. Astfel, în aceste sisteme au fost găsite câteva clase de substanțe active contra cancerului. Deci sunt substanțe cu un nivel de diversitate R1, cu 2 nivele de diversitate R1, R2 și 3 nivele de diversitate. Aceste substanțe, pentru prima dată, au fost testate contra cancer la celule de cancer leucemice, adică la sânge, HL60. Dintre ele s-au găsit mai active cele care conțin inele de benzoice și steroide. În laboratorul nostru, deci, au fost propuse substanțe cu 4 nivele de diversitate, de formulă generală care este aici, cu 4 nivele de diversitate, R1, R2, R3 și poziția Z. Aceste substanțe, deci Z, deci asta înseamnă oxigen, sulf, azot, iar R1 și R2 sunt diferite substanțe, mai bine zis radicali, care formează acest rând. Deci, acest tip de substanțe, noi l-am încercat la mai multe tipuri de celule de cancer, și anume la leucemie, HL60 și K592, la cancer de prostată, în cap, cancerul mamar, MCF7, T47D, cancerul la ficat, HEP, G2 și melanomă, adică cancerul de piele. Deci prima diversitate s-a făcut pe poziția Z. Aici, ca calitate de Z a fost oxigen, azot, sulf. Și dacă ne uităm în partea dreaptă, aici vedem că IC50, asta este concentrația la care mor 50% din celule de cancer, IC50, așa se numește. Deci vedem că cea mai efectivă influență au atomii de sulf, unde IC50 este 9 micromolar, iar la azot 85 și la oxigen 450, adică de la 10 la minus 9 la 10 la minus 4. Se schimbă activitatea anticancer. Aici sunt date aceste date experimentale. Altă diversitate este nivelul 2, când se schimbă poziția R1, de trece cu roșu. Aveți diferiți radicali și dacă observați cel mai activ radical, dacă se păstrează sulful în poziție Z, este piridina. Piridina care conține atom de azot și concentrația IC50 e 10 la minus 7 molar sau 0,1 micromolar. Adică este o activitate destul de mare. Dacă să comparăm cu activitatea doxorubicinii, care este, sau cisplatinii, care este 10 la minus 5, 10 la minus 6. Aici avem 10 la minus 7, adică de 10 ori mai activ. În afară de aceasta, celulele leucemice sunt atacate de radicale când se schimbă poziția R2, nivelul 3 de diversitate, unde am observat că trecând de la radicalul hidrogen, metil, pe urmă benzil, activitatea crește de la 0,1 micromolar până la 0,100 micromolar, adică de 10 ori. Deci, iarăși, optimizarea aceasta duce să venim la concentrații foarte mici, adică ne apropiem de concentrații nanomolare. Și ultimul nivel de diversitate este atunci când noi schimbăm radicalul 3, nivelul 4 de diversitate, ajungem la concentrații de inhibiții IC50 egale cu 3, 3,7, adică 10 la minus 9 este molar, este concentrația în care sunt omorâte sau sunt lichidate în aceste celule de cancer. Prin urmare, aici o consecință importantă este că schimbul radicalului OCH3 în pozițiile orto, meta și para variază activitatea de la 3,7 nanomolară până la 9,9 nanomolară. Iar dacă în poziția respectivă se pune un atom în loc de o metoxigrupă, grupele de ion de brom sau metil sau iod, tot are loc această variație în dependență de concentrație și de natura acestor radicali. Aici sunt date experimentale pentru diferite cancer, cancer prostată, mamar și cancer la picat. Aceste rezultate au fost publicate și în diferite reviste s-au publicat și brevete de invenții care protejează aceste substanțe. Ceea ce am vorbit până acum se referă la substanțele de caracter organic, adică în compoziția cărora nu este altceva decât carbonul, hidrogen, atât. În aceste substanțe au fost introduse și ioni de metal, formarea combinațiilor coordinative, așa zis. În toate aceste cazuri s-a observat următorul lucru, că introducerea ionului de metal, de obicei, ca regulă, duce la creșterea activității anticancer. De obicei, această creștere este de aproximativ de 10 ori, adică îmbunătățește starea activității atunci când ionii de metal se află în compoziția acestor substanțe. Aici avem alte exemple, cobalt bismut, la fel, în care s-a observat că natura metalului care se află în compoziția acestor liganze schimbă și structura pe de o parte. Avem structură monomerică sau dimerică sau trimerică sau polimerică, care la fel influențează asupra activității anticancer. Un lucru ar vrea să subliniez că activitatea ligandului în raport cu activitatea complexului metalic se schimbă destul de frumos. Adică de la, să zicem, avem ligand care are activitate IC50 0,12 micromolar, iar introducerea cuprului în acest complex are activitate de cel puțin 10 ori mai mare decât și la altele ajunge chiar până la concentrații nanomolare. Natura metalului care a fost introdus în diferiți complex, la fel influențează asupra activității, însă pe primul loc rămâne până când cupru și ferul. Dacă ne referim acum la mecanism, care este mecanismul de inhibiție? Deci, în principiu, se observă că lucru care a fost demonstrat prin diferite metode fizice, RMN, raze X, că interacțiunea dintre ligandul care noi l-am construit și ADN-ul se formează prin legăturile de hidrogen. Și natura atomului de azot piridinic joacă un rol important în activitatea acestor molecule. De ce? De exemplu, dacă avem azotul în poziție normală, deci activitatea este cam 10 la minus 6 mol pe litru. Iar dacă schimbăm poziția acestui atom piridinic, atunci activitatea scade dramatic. Asta demonstrează faptul că rolul acestui atom de azot este crucial. Cercetările care au fost făcute cu raze X, deci unde s-au făcut densități electronice pe atomul de azot, a demonstrat acest lucru, că acolo unde densitatea electronică este mai mare, acolo și activitatea este mai mare. Iată, și capacitatea de inhibare corelează cu sarcina pe atomul de azot piridinic și aceasta este demonstrat experimental. În adaos acestor activități demonstrate cu ajutorul razelor X s-au făcut și corelații cu rezonanța magnetică nucleară la azot 15, unde s-a demonstrat că azotul piridinic, cu cât este mai mare densitatea electronică pe el, cu atât este mai mare și activitatea anticancer. Iar odată ce se schimbă această densitate pe azot 15, activitatea scade la 0. Și pentru ultima substanță din tabel, dacă observați, unde este densitatea electronică maximă, minus 3,59, deci activitatea este 100% de toate concentrațiile. Demonstrarea acestui fapt a avut loc prin diferite metode, dar cel mai important este că, într-adevăr, formarea legăturilor de hidrogen dintre ligand, substanța noastră, și ADN-ul este principală în mecanismul de inhibiție. Și acum ne întoarcem la niște rezultate care noi am vorbit mai înainte. Este vorba despre selectivitate. Iată, luăm doxorubicina, deci la celule de cancer mamar și la celule normale. Se vede că este aceeași activitate, adică se omoară în aceeași măsură și celule de cancer și celule deci normale. Adică nu este selectivitate și într-un fel. La următoarea substanță este o substanță de-a noastră. Deci acea selectivitate, dacă observați, indicul de selectivitate 2,6, se socoate că este o selectivitate slabă și la următoarea substanță la fel. Iar la ultima substanță, unde indicul de selectivitate este 12, înseamnă că 12 celule de cancer sunt distruse față de o celulă normală. Deci această selectivitate în terapia chimică, deci se socoate o selectivitate bună. Avem și alte exemple, dar deci nu avem timp ca să ne oprim foarte detaliat. Am vorbit și despre faptul că importantă este și toxicitatea sau doza letală pe care au compus și pe care noi avem. Iată compusul care noi am utilizat în majoritatea cazurilor, dacă ne uităm aici ce scrie, îl dă 50 mg pe kilogram. Sunt clase de substanțe puțin toxice sau cu toxicitate moderată. Adică ele pot fi recomandate pentru utilizare practică în medicină. Din concluziile pe care am putea trage de ceea ce am spus până acum, deci să spun că au fost realizate design inhibitorilor de proliferare a celulelor tumorale de o nouă generație care conțin molecule organice cu setul de atomi, azot, oxigen, sulf, selen, complexe a metalelor non-platinice, ce se deosebesc prin structura geometrică, structura electronică, stabilitate termodinamică și o toxicitate redusă. Peste 1000 de substanțe au fost sintetizate și studiate ca inhibitori ai proliferării celulelor de cancer de tipul HL60, leucemie, MCF7T47D, cancer la glandă mamară, HEMG2, cancer la ficat, RNCAP, cancer la prostată. Au fost selectate clase noi de substanțe biologice active, o capacitate de inhibiție sporită la nivel de concentrații nanomolare. Au fost realizate colaborări în aceste cercetări cu Universitatea de Medicină și Farmaceutică de Timișoara, cu Institutul Național de Oncologie, la fel cu în cadrul colaborării Moldova-România, deci diferite proiecte, și a grantului individual cu Guvernul Canadei. Proiectul internațional ECONET cu tema metodei netradiționale de sinteză a fost făcut cu Universitatea Paul Sabatier din Franța. Și alt proiect internațional cu Polonia, Research and Development of New Anti-Cancer Compounds. Cercetările actuale de perspectivă, deci care se fac acum, sunt coordonate de Universitatea de Stat din Moldova, împreună cu Universitatea din Canada, Universitatea de Medicină de Timișoara și Institutul Oncologic. Colaborări foarte strânse am avut echipa noastră de la Chișinău cu profesorul Donald Poirier, Centrul Oncologic Universitatea Laval, Canada, unde chiar acum se află în deplasare doamna Garbuz, pentru a efectua cu o serie de noi substanțele noastre cercetări oncologice. Și aduc mulțumire la colaboratorii mei, Novitski, Bârcă, Țapcov, Palade, Bulimestru, Graur, Paholnitski, Garbuz, Tatiana Căpățână și Angela Grau. Vă mulțumesc pentru atenție.
Domnule academician, și noi vă mulțumim pentru o prelegere foarte interesantă. Distinți auditoriu, noi avem posibilitate să formulăm întrebări, deci eu vă rog...
Da, cred că de acum se poate face.
Da, deci acum nu este nici barieră de limbă și orice întrebări pot fi, dacă să...
Data trecută au fost întrebări.
Da. Deci, până să mă gândesc...
Doamna Svetlana e prezentă.
Vă rog, doamna vicepreședinte.
Domnul academician, dacă ne puteți spune câteva cuvinte despre testările clinice.
Deci, sintezele care sunt realizate în laboratoarele din la Universitatea de Stat, de fapt, majoritatea cercetărilor sunt făcute în laboratorul de chimie coordinativă. Acum, de fapt, laboratorul și-a schimbat denumirea, este laborator de cercetări biologice, de fapt, și chimice, care sunt îndreptate spre a căpăta noi substanțe. Deci eu am spus care sunt calitățile lor. 1, să fie puțin toxice, 2, să fie selective și 3, să aibă activitate nouă. Deci de activitate.
Da, mai sunt întrebări, da, vă rog, gândiți-vă.
Bună ziua, domnul academician, mă numesc Feodman Daniu, tot absolventul Facultății de Chimie și teza mea era bazată pe sinteza selenosimicarbazonilor, doi acetilpiridini și de asta aș vrea să vă întreb, direcția selenosimicarbazonilor este una de perspectivă? Cum credeți voi, după părerea dumneavoastră?
Da, eu mulțumesc, într-adevăr, în tematica laboratorului nostru, tiosemicarbazoanele și tiosemicarbazidele joacă un rol important și în baza lor, de fapt, s-au format multe alte substanțe cu proprietăți anticancer și eu socot că această este de perspectivă din două puncte de vedere, primul sunt toxice și în al doilea rând sunt o activitate destul de mare. În principiu, la selectivitate încă trebuie de lucrat, dar deci totuși ele sunt de perspectivă.
Vă mulțumim. Mai sunt întrebări? Deci, dacă nu sunt, în primul rând, încă o dată vreau să-i aduc mulțumiri domnului academician pentru această prelegere și vreau să, chiar recent, domnul academician Aurelian GULEA a fost ales membru de onoare al Academiei Române și avem această oportunitate să-l felicităm și să-i dorim succes în continuare. Da, dragi elevi, studenți,
Eu cu
da,
plăcere
spuneți.
vin la fața dumneavoastră și îmi place când sunt întrebări mai multe. Deci.
Bine, domnul academician, întrebări pot fi formulate și mai apoi și transmise. La noi au fost și așa cazuri. Mâine dimineață începem la ora 9:00. Prima prelegere va fi a academicianului Viorel Pisăcaru, Universitatea de Stat, Medicină și Farmacie Nicolae Testimițanu, rolul savanților în descoperirea esenței epidemiilor, o temă foarte, foarte interesantă. Noi am trecut recent printr-o epidemie așa serioasă, eu am în vedere coronavirusul de tip nou SARS-CoV-2, așa că aici o să fie un lichbez așa ce înseamnă epidemie și în general cum ele, parcursul istoriei, au influențat. Și ați văzut astăzi, domnul profesor Rotman a prezentat acea dependență. Eu cred că ne pune pe gânduri, de fapt, dependența pe perioade medii de viață, spunem așa, în funcție de timp. Și ați văzut că ea a fost la un nivel foarte scăzut și pe urmă brusc a crescut. Și brusc a crescut atunci când știința a început să se dezvolte. Și într-adevăr, dacă nu era știință, era cu toată altă situație pe mica noastră planetă. Deci, vedeți că noi acum toți ne folosim și din tehnologie și avem posibilitate să interacționăm cu cunoscuți, prieteni care sunt în diverse părți ale lumii. Deci, și lucrul ăsta a devenit posibil grație dezvoltării științei. Și o să aveți posibilitatea zilele acestea să ascultați prelegeri care să fie prezentate de experți, mari experți în domenii concrete. Încercați să luați maximum din aceste prelegeri. Mâine majoritatea vor fi în limba română, dar vor fi și prelegeri în limba engleză. Eu repet, chiar și asta trebuie să fie un training. Un training pentru că una e să poți ști în engleză și alta e să la auzi, să prinzi, să înțelegi ce vorbesc, cu atât mai mult că bine vorbesc persoane de peste hotare. Astăzi profesorul Rotman din Statele Unite, așa, limbă perfectă engleză, dar desigur că deci, trebuie să fie bine antrenat ca să înțelegi la auz și să prinzi. Dar aceste prelegeri vor rămâne pe site-ul Academiei de Științe a Moldovei și chiar în viitor puteți să vă întoarceți să mai ascultați unele fragmente, să încercați să intrați în esență, cu atât mai mult că domnul profesor James Rothman va veni în Republica Moldova și atunci poate veți avea posibilitate chiar să formulați întrebări și să discutați la direct. Noi vă felicităm cu ocazia deschiderii școlii de vară, deja 3 prelegeri au avut loc și vă așteptăm mâine la ora 9, deci prima prelegere. Da, o fotografie de grup, idee foarte bună. Veniți toți în față aici și o să facem fotografie de familie, pentru ca să rămână în istorie.